In the part 2 of this lecture, we will see how can we convert the proton motive force stored in the intermembrane space into ATPs. At the end of the electron transport chain, we have built up a high gradient of proton in the intermembrane space, and it has a lot of potential energy like a dam that is ready to release its water, and we can harness that potential energy by converting it into the kinetic energy and eventually electrical energy by using a turbine. And guess what? We also have a turbine in our mitochondria, and that turbine is called an ATP synthase. So due to the high proton gradient in the intermembrane space, these protons will eventually rush back to the matrix by flowing through the ATP synthase. And it is the motion of this proton that propels the ATP synthase to add a phosphate group to an ADP, forming an ATP molecule. Okay, showing in this structure, you have an ATP synthase, which is a beautiful piece of molecular engineering. Please note that in this picture, it is upside down as compared to the earlier diagram. So over here, you have the matrix, and here at the bottom would be your intermembrane space, where you have a high gradient of protons. And as you can see, ATP synthase is a giant complex composed of many protein subunits. And the yellow part of the ATP synthase is static, while the green part is composed of protein components that are rotatable. And remember, we have got a high proton gradient over here, and they're trying to get over into the matrix. However, the inner membrane of the mitochondria is impermeable to ions such as the proton. So the only way that the proton to get into the matrix is by going through the ATP synthase. But how can the movement of protons make ATP synthase turns? Well, here comes the genius design of the ATP synthase. First of all, it has a subunit A, which contains two half channels, channel 1 and channel 2, one facing the matrix, which is channel 2, and channel 1 facing the intermembrane space. So the protons will first enter through the channel 1 and try to get out. But the only way to get out is through subunit C, which has the negatively charged aspartic acid attracting the positively charged protons. So when proton bind to subunit C, it neutralizes the charges on the subunit C, which makes it lipophilic and allows the C ring to rotate through the hydrophobic region of the lipid membrane. And when it arrives on the other side, there is an opening for the proton to flow into channel 2 and eventually release back into the matrix. So the rotation of this C ring is transmitted by a gamma subunit to the hexamer, which then changes its conformation and allows it to generate ATP. Well, I think you can understand it better by watching the video which I posted on Canvas. It is an old video with very poor quality graphic, but anyway, I think it has summed up the mechanism really well. By now, you should clearly see that it is a transfer of electrons along the electron transport chain that causes the conformational changes in protein complex 1, 3, 4, which pump protons across the inner membrane into the intermembrane space, and this has built up a proton gradient that allows ATP production by using ATP synthase. Okay, here is a quick question to test your understanding. In the electron transport chain, oxygen is fully reduced into what? Yep, the answer is water molecule. Now, on the other hand, partial reduction of oxygen will produce ROS, or the reactive oxygen species, such as the superoxide ions and peroxide ions, which can lead to aging and many other diseases. However, the good news is that these molecules or these ions can be removed by specialized enzymes, such as superoxide dismutase that can remove superoxide ions, or catalase that can remove or that can degrade the peroxide ions. Now, for information, ROS is a natural side product of metabolism. It happens at all time, but at very low controllable level. But during times of environmental stress, such as exposure to UV and heat, then ROS levels can increase dramatically. To digress a little bit, here is an example of aging caused by UV exposure. So this man has been driving his truck for more than about 28 years. And because of the constant exposure to sunlight on one side of his face, so this side of face has so much wrinkles as compared to the other side. Again, this aging process is caused by ROS, the reactive oxygen species. And judging from the site of the damage, can you predict which country did he come from? Well, he's coming from USA, as the driver's side is on the left. Okay, you have seen the importance of oxygen in the oxidative phosphorylation because it acts as the final electron carrier in the series of electron transport chain. This is why we need hemoglobin to transport oxygen in the blood and myoglobin to send this oxygen into the muscle tissue. But we can also intoxicate the system by having carbon monoxide, which is analogous to carbon dioxide, but it binds 400 times as stronger with hemoglobin. So even though we have a tiny amount of carbon monoxide in the air, it can still suffocate us because it will take away the oxygen binding site in the hemoglobin. Cyanide, on the other hand, is even more toxic, which can kill us in minutes if not seconds. Why is it so toxic? Because it binds to complex 4 irreversibly and blocks the whole process of oxidative phosphorylation. As a matter of fact, you and I have only ATP to sustain for about 2 to 10 seconds. And as a result, we are regenerating new ATPs every moment. So when cyanide shuts off the system, we can only survive for about 2 to 10 seconds. Mitochondria is one of the very rare organelles which contains its own DNA and is known as the mitochondrial DNA. Therefore, mutation might occur and it could lead to mitochondrial defects. One example of mitochondrial defects is the leaky membrane. So when you have a leaky membrane, you tend to generate less ATP than the normal people. And this partially explains why people with the leaky membrane can eat a lot of food, but they gain weight slowly as they generate less ATP for the anabolism of fats and proteins. And of course, it also depends on the type of diet and physical activities. So having a healthy lifestyle is always more important. Okay, by now I believe you have a complete idea on the metabolism of glucose molecule. First of all, glucose must be imported to the cell via the glucose transporter protein. Then it's broken into two pyruvate molecules along with the production of two ATPs and two NADH. So this process is called glycolysis and it does not require oxygen because it is an anaerobic reaction which takes place in the cytoplasm of the cell. Then the pyruvates are decarboxylated to become acetyl coenzyme A, which is a 2-carbon compound. Again, you have the release of 2 NADH and 2 carbon dioxide. Then the acetyl coenzyme A will bind with a 4-carbon compound from the Krebs cycle, which is the oxaloacetate. And in this reaction, you have the production of 4 CO2, 2 GDP, 2 FADH2, and 6 NADH. Finally, all these high energy electron carriers are being oxidized in the electron transport chain, which is the oxidative phosphorylation. And it's from this reaction you get ATP production. So always remember, this reaction will require oxygen because they are the final electron carriers in the electron transport chain series. And where is this reaction happening? It happens right in the inner membrane of the mitochondria. And for the Krebs cycle, it happens in the matrix of the mitochondria. It is amazing that our nature has been in balance for thousands of years due to these two important organelles, which is chloroplasts, they undergo photosynthesis, and mitochondria, they undergo respiration. So chloroplasts convert carbon dioxide and water into oxygen and glucose through photosynthesis, while in respiration, we reverse this process by oxidizing glucose back into carbon dioxide and water molecule. And with that, we have got the release of energy to power the formation of ATP. In your PowerPoint, I have attached two videos which I believe could be helpful to your learning, such as this one and this one. And with that, we have come to the end of this lecture.